As Adam said, my name's Clare Bernard. I run a group called the Data Sciences Platform at the Broad Institute. It's a group of about 250 people, mostly software developers who work on open source software that supports bioinformatics research, and that includes GATK as well as Terra, which is the main topic for today. Most of those software developers are people with kind of industry backgrounds, but who are really, interested in working in the nonprofit space and interested in having an impact in life sciences and in research in the life sciences. So today I'm going to talk about data in genomics, as well as the problems we're trying to address with our work, and a few of the specific scientific problems that we're working on. So since the Human Genome Project was completed in 2001, a lot has changed. Genomics, which once could have been considered radical, has become really mainstream. It's now considered fairly normal for people to do carrier screening, have genetic screening done during pregnancy to look for chromosomal abnormalities. We've got large databases mapping the sources of monogenic disease. We can calculate polygenic risk scores. We have N-of-1 therapies. There's really been a ton of great discoveries. And we've seen the cost of sequencing really go down dramatically, which has made it easier to sequence larger and larger populations of people. If you're trying to unlock the mysteries of human genetics, today there's a lot of data to work with. Vast amounts of genomic files, as well as phenotypic data, medical records, imaging, as well, and it's not just genotypes, arrays, exomes, single cell data, spatial transcriptomic data. And we've seen governments and other institutions investing millions of dollars in generating data sets, like some of the ones that are shown here on the slide. Like, for example, the 1,000 Genomes, which has 3,000 participants, UK Biobank, which has 500,000 participants, and the All of Us research program, which I'm going to talk about in more detail later, which has 1 million participants. And you can kind of see like the size of these data sets has scaled up pretty significantly over time. And computing advances mean it's possible to turn this data into really meaningful insights, the kinds of insights that can really improve people and have a dramatic effect on people's lives. However, while all of this data is really, really wonderful in theory, there's a lot of challenges with working with very large data sets like this. When you're working with large data sets, it's hard just to find the data that you want, the particular data that you need for your study. It's hard to download and store the data. It's very expensive to download and store the data. It's hard to interpret and analyze file formats. There's a lot of different formats, and some can be confusing, especially if you're combining data from large data sets that might use advanced compression. It's hard to share data with collaborators in a way that's efficient and secure. Data security is a massive issue. A lot of this data is very, very sensitive. And so making sure that we're storing it in a way where the data is used responsibly and used for in a way that's consistent with how the data was consented is a challenge. Methods of granting access to the data is really cumbersome. So here on the right, I'm showing a plot of data access requests that have been submitted to, this is data from dbGaP, so data access committees for that data that's in dbGaP. And you can see that it's going up kind of exponentially over time. As we have more and more of this data, as more and more great research is being done, more and more data access requests. And right now, the process for giving access to researchers is really, really manual. And so it can take a long time for a researcher to get access to data. And many times we see researchers just give up on getting access to the data and pick a research project where they already have access because of these challenges. Integrating data sets is really hard. If you've got the same type of data, but it was generated in different places, you can have batch effects. Phenotype curation is really manual and really difficult. Integrations across data types is really hard. And analysis comes with a lot of challenges as well. So it requires really large, expensive infrastructure investments to handle the time when you need a lot of processing power, even though most of the time you don't necessarily need all of that processing power. And anyone who can't afford the big infrastructure is locked out, making genomics kind of a sport of a sport of kings, you know, only possible for people who are at institutions that have really large infrastructure. Analysis tools and computing environments are also often complex and custom built. It's hard to find the right tools and set up the right computational environment, which is especially true if you're not a programming expert. And these complex custom analysis tools and environments can lead to non-reproducible results, which makes it even harder to build on existing studies. So we kind of had this first challenge of genomics, which was generating the data, generating the first human genome, making it cheaper and possible to generate these really large data sets over time. And I think we've now reached the point where we have a second challenge, which is really about data science and data technologies. And this is where I think open source software can play a really big role. So the first opportunity that I see here is to bring researchers to the data. And this is kind of the first big shift that I think we need to make as a community. Traditional bioinformatics looks more like the picture on the left, where you have a central repository that holds the data, and then each researcher copies it to their local storage to analyze it with their own analysis tools in their own custom computing environment. There's a lot of negatives to this model. For one thing, you're copying the data over and over and over again, not actually sharing the data. And that creates really, really high data storage costs. It can also create inconsistencies in the data that you're looking at. So that can also lead to non-reproducible results, as I mentioned before, because every analysis is done in an isolated environment using those custom tools we just mentioned. Huge infrastructure costs for expensive fixed compute resources that some can't afford. And finally, it's really not secure, because if you have all of these copies of the data in different infrastructures, your data is only as secure as the weakest security implementation anywhere there is a copy of the data. And all of these things get in the way of being able to actually use all of these great data sets that are coming online. If we move to more of a cloud-based model, and that's the picture here on the right, it looks more like this. You have data that rests in a central repository in the cloud where researchers can access and analyze the data together in a virtual environment. And with this cloud-based data storage, you get true immediate data sharing, where you have one copy of all of the data that all researchers can access and search and analyze together in one place. You only have to pay for that single storage instance of the data. You can have shareable tools and containers, so results are reproducible. You've got cost-effective, elastic compute, so you don't need to pay for compute power when you're not using it. Cloud resources can scale to meet your needs, and you don't need to make big infrastructure investments to build up, big on-prem data centers. So that makes the research more equitable as well. And finally, you have centralized data security, which removes the responsibility from individuals and enables comprehensive security fences. At the Broad, we actually made this shift around the same time my organization was born. And we were actually pushed to make this change because we had an on-prem data center. We were doing a lot of sequencing, and our on-prem data center filled up, and we actually didn't have room to store all of the data. And this was a huge risk to the Institute. We were at risk for having to stop production sequencing, and so we had this huge rush to very quickly move all of our data storage, data processing, and everything to the cloud. And so because of that, for the last 6 years, we've been really focused on the cloud-centric approach, and it's really the only scalable way for us to manage the kinds of production processing and production storage needs of a big sequencing center like ours. And I think I showed on the previous slide just how the Broad genomics numbers have been scaling up over the past 10 years. You can see it's also kind of this exponential scale with the amount of data that's being created. And on-prem data centers just don't scale that way. The next big shift that is needed, I think, is around how we grant access to the data. So I talked previously about the number of data access requests. And we actually went through an effort at the Broad to look through all of the consents that we had to see, was it possible to come up with codes to create kind of machine readable consent codes, and came up with the answer that in most cases, something like 95% of the time it actually is, and the consent is pretty understandable. And so this led to the creation of the GA4GH data use ontology that's shown here. And that gives us the ability to then to use these machine readable consent codes to algorithmically match someone's research purpose with the data that is properly consented for that particular research purpose. And I'll talk about that a little bit further on. But I think one really important point here that I want to keep emphasizing is that security and access to data doesn't need to be a trade-off. It can be, obviously, if you have a data set that's on a computer that is locked in a box at the bottom of the ocean, it's probably very secure and nobody can access it. But there are ways of very securely granting access, making access to data for the proper research purposes really, really feasible. This third one is really the one that's kind of maybe the most moral and ethical. It doesn't get more personal than your genome. And I think for the benefit of patients and research, we have an obligation to be responsible custodians rather than think of ourselves as data owners. It's the patient's data, not ours. So that means for groups like mine, where we manage these large data sets, we should think of ourselves as custodians, and we're responsible for making sure that that data is used in a way which is consistent with how the patient or the patient community wanted it used and how it was consented. And I think direct to participant studies are a really interesting model here for how this can work. And we've seen really good outcomes when you run studies this way. So in this model, rather than having researchers collect data by going through a medical center, you can actually have disease foundations recruit the patients that have their particular disease, use a software platform to collect that data, and you can get the researchers and patients talking much more directly with the disease foundation, still ultimately representing patients as owners of that data, rather than the researchers or the software company. And just as a success story and an example of this, this was one study that we ran, collaborating with TIMU and the Ataxia Telangiectasia Disease Foundation. And so we built out a software platform for this study. Or we have built out a software platform to run studies like this, and this was one of the studies for it. This study recruited over 400 children with AT across 12 languages and 39 countries, performed whole genome sequencing, and very importantly, all of this data was owned by the patient community. TIMU, who's a prominent researcher at Boston Children's Hospital, analyzed the data and actually realized that the therapy might work for 26% of the patients. And this resulted in a clinical trial being initiated last year, which I think is really exciting. So going a little bit further into the software that we develop, the Terra Platform is one of our largest efforts, and really is kind of underpins a lot of the other software efforts in our group. And my group, as I said previously, the Data Sciences Platform develops open source software that is really focused on some of those challenges that I talked about previously, and kind of pushing the field in that direction. Terra is developed as a collaboration between the Broad Institute and Microsoft and Verily. Terra is also the platform that we use for all of our own processing and storage. So this is where we store all of the data that is generated at the sequencing center, process all of the data that is generated at the sequencing center, as well as by other Broad labs. This is also how we manage access to that data, how we share the data. And then it's also a web interface, which is designed to do the heavy lifting and of analysis, where researchers can come and do their own analysis or move their analysis from on-prem infrastructure to the cloud. So some of the design principles that we used when creating Terra. Open source, modular, standards-based, and community-driven. These principles really came up while we were working on a series of projects with a bunch of different collaborators at other institutions. And what we realized was that for all of these projects to be successful, we all needed to be able to create an ecosystem rather than monolithic silos. And so in order for us to create an ecosystem, we needed to each be able to create software components that, that could interoperate, where one institution could store the data, another institution could create software to analyze it, another institution could create tools for it. And we kind of saw this model over and over again with projects like the All of Us Research Project, which I'll talk about a little bit more, the NCI Cloud Commons, the NHLBI Biodata Catalyst, the Human Cell Atlas, NHGRI Anvil. For all of these projects, you have maybe 4 or 5 different institutions that are collaborating together on the project. And in order for that to really be successful, all of the software needs to be built with these kind of principles in mind. And we kind of called this concept, when we were thinking about it, the overall concept of the data biosphere. We know that not all genomic samples are going to be sequenced at our sequencing center, but it's important that you can be able to analyze that data with data that's generated at other sequencing centers. So standards become really important. Similarly, we know we won't host all important genomic data sets in Terra or create all of the interesting tools. So we want an infrastructure that researchers can really easily plug into. So what is Terra? These are some of the core capabilities. So one of the really important pieces of functionality is the data library. So a lot of data sets, and I'll go more into which ones specifically, but a lot of data sets are available in the Terra data library, and that's where researchers can come search for what data sets are available. And we're building out a lot more capabilities around searching for specific things related to subsets of samples across different data sets that make it really, really easy to find the data that you need for your particular research project. We also have this concept of workspaces, where a workspace is kind of a collection of the data that it's the kind of sandbox for your particular analysis project. And in that workspace, you have the data relevant to that analysis project, which is just a reference to the data, since we're not actually copying any data anywhere different. You have the tools that you're using for that particular analysis project. And it's a secure and shareable unit and shareable environment. So you can add collaborators to your workspace. Once you've finished an analysis project, you can actually publish the workspace and make it visible to others, which then is really great for reproducible research, because you can show others exactly what you did in the platform. And you have complete control over who can see what. We also have workflows. So we have our own workflow engine. And this is where we also have a library of workflows, including all of the Broad production workflows that we use for processing our own data at scale, and then workflows that others have added. We also have a connection to DocStore, the UCSC DocStore, which has many other workflows that have been added by other researchers, so that you can easily add those to your workspace and run them on your data. And then we also have other additional interactive analysis tools. So, so Jupyter Notebooks is built into the platform. Our Studio, Galaxy is a recent addition. And so all of those you can use to analyze your data still in that workspace environment, in that secure workspace environment. And it's super important to us that we're making it really easy and cost effective to manage and analyze these very large data sets in the cloud, simplifying access to this data while keeping the data very secure and acting as custodians of this data rather than the owners. So, so as I said previously, Terra is really part of an ecosystem, an open ecosystem, not an island. You can find and access data, but you can also bring your own. You can access tools, but you can also bring your own. You can perform analyses and share them securely. So you have a lot of flexibility. It's also built for multi-cloud. So currently, most of these capabilities are available on GCP only, but we're building out support for Azure, and you'll see a lot more of these capabilities show up on Azure over the course of the next year. So this is kind of a diagram showing how some of those components that I talked about previously are laid out. So you have external data repositories. These are data repositories run by other institutions that we collaborate with, as well as data that is stored in Terra itself. You have workspaces where you can connect to our own Terra data repository. You can import your own data, which can just be uploaded directly into the workspace. The workspace has its own storage. You can also connect to those external data repositories. And then from the workspaces, you can access these other analysis tools that I mentioned previously. And you can import tools from GitHub, DocStore directly, or upload your own tools. And then you can also connect from data that is in Terra to other analysis platforms if you would prefer to use other analysis platforms. And then on top of all of this, so the kind of primary interface is the Terra Community Workbench, but we also have other applications built on top of the same core platform. So the All of Us research program has its own workbench and its own interface that is specific to that data set. We also have portals, and one good example of this is the single cell portal, which also runs off of the Terra platform, but is aimed at kind of specifically at visualizing single cell data. So there's kind of a narrower use case, but is much more. It's a little bit more kind of point and click, and has a lot of really interesting single cell specific visualizations. And then you can search for single cell data in single cell portal, take some of that data back to the workbench, and run further analysis on it. Here's some subset of the data that is available in Terra today. Some of these projects I mentioned, Human Cell Atlas, a lot of that data is available in the single cell portal too, NHGRI Anvil, All of Us research program, TopMed, NCI, Target and TCGA, Biodata Catalyst datasets, Nurses Health Study. Some of these data sets are stored in Terra itself. And as I said previously, some of these data sets are stored by our collaborators, but can be accessed and analyzed in Terra. Centralized security. So, as I said previously, when you have this cloud model, you're really creating a much more secure environment for the data and able to protect the data better. So centralized security means all components in the ecosystem exist within a FISMA and FedRAMP moderate environment. And this satisfies the security requirements for many controlled access data sets. Terra is a FedRAMP authorized Terra is FedRAMP authorized as a moderate impact system with agency authorization. You can find Terra listed in the FedRAMP marketplace. I think it's really important to emphasize that open source does not mean open access and does not mean lack security. It's very possible to build a secure open source system, and this is really important for a lot of the data sets that we host and a lot of the projects that we work on. and something that we take very, very seriously. So thinking a little bit more about that second challenge that I mentioned around how we can simplify granting access to data. Duos is a separate project, but is integrated with Terra. Duos stands for the Data Use Oversight System. This is based on the GA4GH data use ontology. And I really think of Duos as being three things. The first is that it's interfaces to transform data use restrictions and data access requests into machine readable code and into that GA4GH Duo standard. The second thing is it's a matching algorithm. So it matches that Duo code with a research purpose. And it also provides software and interfaces and a UI for data access committees to use to adjudicate whether structuring and matching has been done appropriately. So it enables data generators to deposit their data and kind of register their data sets with the system using those consent codes. Then a data access committee can view what the results of the Duos algorithm recommends, and this can speed up their review process. Data requesters, it can help structure research purposes so that then requesters can easily check what data they are allowed to access, and a lot of that is the part that's really helpful in Terra, where in the Terra Data library, a researcher can then search for what are the data sets that have a matching consent for my particular research purpose. So Duos doesn't replace anything that the Data Access Committee does, but we have found that it can significantly speed up the work. And in order to kind of answer that question of how much can we trust to the Duos algorithm and how much can we rely on the Duos algorithm, and rather than have everything be manual and done by the Data Access Committee, we've been running a series of pilots with the NIH. So we're currently working on running this pilot with the NHGRI. The NHLBI went through kind of phase 1 of a pilot where they reviewed test data access requests in the Duo system and are interested in participating in the bigger pilot. NCI has expressed interest. There's GA4GH driver projects, including Australian Genomics and Autism Speaks, that are planning on testing Duos this quarter. And we have a data access committee at Broad that is actively using Duos to receive and process data access requests. And all of this pilot activity is really aimed around answering these questions of, how well does it work to translate a data use letter to structured data use limitations? As we go through the data access request review, how do we evaluate these structured terms with an algorithm to make the same decision a human data access committee would make when reviewing a data access request? And what are the tricky cases where we really need the human, more manual review? And then finally, how do we translate a research use statement into a structured research purpose? So now I'd like to talk about two of the projects two of the scientific projects that we're using Terra for, and where we're having a really interesting impact. So first, I'll talk about the All of Us research program. As you saw in kind of one of the first slides, this is a really interesting, really cool data set. This is an effort to engage more than 1 million US research participants. And there's really a huge emphasis here on recruiting a really diverse group of participants, which is really important. And in this project, we're sharing tissue samples, genetic data, lifestyle information, electronic health records, and pioneering a new model of research that emphasizes engaged research participants, responsible data sharing, and privacy protection. As I said, all of us puts a ton of focus into getting a really diverse set of participants. And here you can see the community and provider partner network. This is a little out of date as of January 2021. And all of us is a really big project. The Broad is part of the Genomics Center, so we do some of the sequencing for the program, and we're also part of the Data Research Center, the DRC. And so here I'll be focusing on the Data Research Center, which is what Terra and what our software supports. And for that Data Research Center, this is a collaboration between Broad, Vanderbilt University, Vanderbilt Medical Center, and Verily. And the DRC is really responsible for hosting all of the data and then providing access in a researcher workbench to the data so that researchers can analyze it. And that includes all of the curated phenotypic information as well as the genomic data. So the DRC is responsible for security and privacy, 3 tiers. So the data is kind of organized into controlled tier data, registered tier data, and public tier data. Then there's a raw data repository where we store all of the data that is generated as part of the program, and a curated data repository, which is after we've done a lot of work to make that to harmonize the data, curate particularly the phenotypic information, and make that available in a more useful form to researchers. So all of this is part of that data and research center. There's also a public data browser, so that's where you can see the very anonymized high-level statistics that that can be available to look at by the public. Genomic data, and then researchers can access registered and controlled tier data through the researcher workbench after going through some initial training and security checks. So as I mentioned, all of us has a public data browser, and which means that potential researchers can actually browse the available data and before deciding whether to register. So I'd encourage you to go ahead and look, and you just go to this URL right here, and you can look through all of the data that's available as part of the program. It's databrowser.researchallofus.org. And registration is easier than most other studies at this magnitude. So we've really tried to make it secure, but as easy as possible to register and start looking at the data and start working with it. So as I mentioned, Terra supports the researcher workbench. So the researcher workbench has a lot of the same concepts that I described earlier, where from the researcher workbench, you can search the data. There's all of us phenotypic information is organized into the OMOP format. And so you can use, so there's a data browser that is specifically built for that data format. So you can use that to select subsets of the data. But it still has this concept of workspaces where you go to a workspace and you can access certain tools, reference the data set, reference the data. And there's a lot of research that's already currently underway. So right now it's mostly the health records that is in the platform. We're adding genomic data this year. Some is already added, but there's more coming. There's a lot of active projects. We're already starting to see publications. Lots of conditions being studied. Lots of researchers from many different institutions around the country, and we're seeing some really great results already. All of us is also an interesting example of a project with very strict security requirements. And because of a lot of the things that I mentioned earlier around security being easier to do when you've got one copy of the data in the cloud, it's also an example where no exfiltration is actually allowed at all. So you're not allowed to download the data. There's a careful balance there, where we want researchers to be able to download figures that have been generated in the platform, for example. So downloads like that obviously are allowed, but we monitor very carefully any data that is leaving the platform. And there's a lot of security software infrastructure set up to make sure that the data is as protected as we can make it in the platform without getting in the way of good research getting done. So this is a little bit out of date as of May 2021. This is all of the data that is currently available in the Workbench. We've got 263,000 physical measurements, 203,000 electronic health records, 8,000 Fitbit records, lots of surveys that capture participant data like lifestyle, healthcare utilization and access, COVID data. We have 316,000 participants already. Again, the goal is to get to over 1 million. And 80% are from underrepresented communities, including 50% racial and ethnic minorities, which, as I mentioned, is really a focus of the whole effort. So there's already a ton of great data and a lot of really good research happening. And you can go to researchallofus.org/spotlight and see some examples of the really interesting research that's being done on the platform already. So the second project that I'd like to talk about is one that's near and dear to my heart, particularly since I'm giving this talk from my home. And obviously, the whole pandemic has had a huge impact on all of us over the past couple of years. And it's crazy when you think back to now almost 2 years ago, everything that has been done, and how our mindset shifted about how we were thinking about this data and what we were doing to fight the pandemic. So back in March 2020, the Broad Sequencing Center had started scaling up a really big testing program. And in parallel, the Broad Viral Genomics Group, which does viral sequencing separate from the sequencing center, started sequencing SARS-CoV-2 samples from MGH. And this is a group that the data sciences platform already works with a lot. And so as they started sequencing these samples, we worked with them to establish these analysis methods in Terra. So we were processing this data in Terra, and doing a lot of the analysis of that data in Terra. And that became really important going forward because then in August 2020, we started getting information. Again, these were initially just samples from around the Boston area, but we already started finding, having really interesting findings from around super spreading events that were happening, and it became really clear the impact that very large conferences had on the pandemic. And one of the great things that Terra was able to enable here was, because all of this analysis was already happening in Terra, it was really easy to package up those workspaces where the analysis was happening and all of the methods and data that were being used, and just make those publicly available so that others could, if they had different data, they could upload that data and run the same exact analysis methods, or they could just come to see the analysis that we were doing that was resulting in these findings and these papers. So the Broad Viral Genomics Group continued sequencing through the end of the year. In January, we got a new administration took office, and there was cases were still really, really high, even higher after the holidays. There was a need, and there was more funding for larger scale surveillance efforts. Because we were already doing all of this testing in the Broad Sequencing Center, it was very easy then to start scaling up sequencing. Well, I shouldn't say easy. I think the people in the sequencing center would certainly disagree with that. We were maybe at an advantage there, but they actually had to, the sequencing center hadn't done viral sequencing previously. And so in order to scale up this effort, they actually had to, rent additional space, and there was a big effort around scaling this up. So we scaled up the sequencing effort, both with the sequencing center, the viral genomics group, and the data sciences platform, and made all of the data and pipelines available in Terra as well. So this is an example of some of the impact that all of this had. So February 2020, I mentioned the analysis where where there's a conference that was this super spreading event. So this was a key example where having all of this data and being able to make it available in Terra was really, really great. Helped us understand the impact of very large gatherings in the pandemic, and our software was able to lead to very transparent, reproducible analyses. And I want to acknowledge some of the kind of key researchers who participated in this work: Pardees Sabeti, Bronwyn McInnes, and Christine Loreth. And as we were doing this, we were partnering really closely with the CDC. So as we continued sequencing all of this data, continuously monitoring this data and monitoring the variants continued to be really important. And there was a really important kind of time-sensitive nature here. So U.S. public health labs all of a sudden were under all of this pressure to sequence samples at unprecedented volumes, somehow analyze and return this data really quickly. A lot of these labs didn't necessarily have bioinformatics pipelines or large bioinformatics teams. We didn't have a national infrastructure or platform for doing this across the country. But the government was really motivated to do this. We had funding available, and the government was motivated to approve the use of the cloud and platforms like this. A lot of you probably remember the outbreaks in Providence. That's kind of an example where being able to do this analysis really quickly was super important, because when that first happened, we didn't know whether that was because the Delta variant, because there were some breakthrough infections in Providence. We didn't know whether that was because the Delta variant was more transmissible, or whether that was a sign that there was something worse going on, some new variant. And so that was a case where quickly sequencing this data, quickly talking to the CDC about it, was able to result in better general guidance about how we should be reacting to the pandemic in the moment. And then we've continued to partner closely with the CDC. As I mentioned, there really wasn't a national platform for this, and particularly with infectious disease work, the cloud and a platform like Terra are really important because of the distributed nature of the work. So this is a map here on the right showing states where there's either a state public health lab or a county public health lab that is currently using Terra. I think this is actually a little bit outdated, so it's even a little bit more of the country than this. And so we partnered closely with the CDC to develop and support Terra as a general, and are still partnering closely with the CDC to support Terra as a general purpose pathogen genomic compute platform for all US public health labs. And this provides a secure compute environment for access to the CDC recommended analytical workflows, gives these labs the ability to easily share tools amongst labs and share data. And because of this concept of kind of featured workspaces, we're able to create workspace environments that already have these analysis tools, so that people in those labs just need to upload their own data, and then they can run the exact same analysis that is being run elsewhere, and share those results. And this is I think an important model when we also think about what's needed for other kinds of infectious disease, like for example malaria, where it is global. It's a global problem, and you want to enable local groups to add their own data, run analysis, retain ownership of that data locally, but still be able to share analysis methods and see aggregated results all in one place. And this is something which a distributed platform like Terra, using cloud technology, really helps. So there's other interesting projects we're involved with. For example, Anvil is a project where we're actually collaborating with the bioconductor team. There's also the Human Cell Atlas project. But I think the main things that I really want you to take away from this talk is that, all of the challenges that I mentioned at the beginning are surmountable, and open source software and data technologies have a really key role to play. And in fact, many of the current efforts in flight, I think, will be able to really help us overcome a lot of these challenges. And so I'm really excited about what we're going to see in the future of genomics as a result.
Thank you so much, Claire. This was a really great talk. And we've already got first question from the audience, so I'll just move straight to it. And in fact, the question is interesting to me personally, because actually during my preparation for this, I asked that question myself and I Googled a little bit around, and I think what I found was that the answer was no, that essentially if you want to use Terra or you want to run it yourself, it'll be just too difficult to do without kind of direct support from the broad or kind of being involved. So do you have any comments or sort of pushback here?
Yeah, so the short answer is no, but there's a longer answer here. So the answer on AWS is kind of more clearly no. A lot of what we've done is more GCP specific, and we're currently focusing on building out Azure capabilities, but it doesn't currently run on AWS. It is possible for parts of Terra to run in more isolated GCP accounts. And in fact, the way Terra, I should have said this earlier. So it's a software as a service platform operated by the Broad, and we don't charge any researchers, academic researchers, for its use. So the way that it works is you can use your own GCP billing account, and so any of the work that you do in the platform in a workspace and the data that you're storing is all the same as if you were just using the cloud directly. It's just pass-through billing, and so you would get your normal GCP bill in the same way. So you can kind of bring your own GCP account in that way. But you also can deploy parts of Terra locally. So our workflow engine Cromwell can run locally, and you can also deploy the data store component. It's still not trivial to set it up on your own. Like you would have to kind of come talk to us, and we partner with you on helping set it up. But that data store component is something that can be run and operated locally and still connected in with the rest of the ecosystem. But a big part of what we're trying to do is not create silos, but to create kind of this connected infrastructure. And so, and for a lot of this, it is complicated to operate and run, and so it's easier to kind of partner together on it and let us do it once. But if there are those components that you really do want to run and operate locally, that's possible as well in some cases.
Yeah, thank you, Claire. That's a great answer, and actually, there was a bit of a depth to it, as you mentioned. It wasn't just a simple no. One of the things that really struck me when I was listening to your talk is that you really do have just a phenomenal understanding of the product. You were able to bring up such details as what sort of data sharing codes the platform uses, and I'm wondering, historically, or maybe the kind of more traditional way for these type of big leadership roles that you have that you have at the Broad is sort of more oriented around securing funding and talking to stakeholders and sort of like resolving various sort of like interpersonal challenges within the team. But what you're bringing here is just this like wonderfully fresh perspective of just having really great understanding of the product. I wonder, like, how do you manage to do your job, which involves things like funding, things like managing the team, but like also keep this really in-depth knowledge of the product?
Well, thank you. That's very nice of you to say. So my background was originally in particle physics and data analysis, and then I went into software and tech. So I also just have done I learned to program during my PhD when I was studying particle physics data at the Large Hadron Collider. So I learned to program very poorly. I would not say that I'm good at writing code, but I then went and worked for a software company, Tamer, where I was working, very closely alongside all of the developers. And so I do have a pretty strong kind of software background. And I came to the Broad about 3.5 years ago, and initially came not to run the group, but to run the product management function for the group. And so I really spent the last 3 years thinking a little bit less about funding and external partnerships, and thinking pretty deeply about the product and what exactly we were building, and how we could make it better suit the needs for researchers. And so now, I've been responsible for running the group now for the past year, and so, but I still have some of that knowledge from working, more closely with the development teams.
So in short, the answer is we need more leaders with particle physics background and sort of technical expertise.
Definitely, I think that's the way to
go. Okay, wonderful. Another question I have is about UK Biobank. So one of my projects back in my sort of PhD student role at Glasgow, I was actually trying to analyze UK Biobank, and it's a wonderful resource. It's massive. It's especially with the recent exome release. I'm expecting just incredible things when it comes to precision medicine, when it comes to things like sort of multi-feature predictors of cardiovascular disease, etc. But it's very difficult to analyze. It's terabytes of data. And if you were to do it, you can't even realistically do it locally, because even just downloading all that data is going to basically be a PhD project at that stage. So the question is, my question for you is, could Terra be of help here? And is there anything else? Is there anything that we could expect on the kind of UK Biobank front?
Yeah, that's a great question. So, I mean, I think UK Biobank is similar to the All of Us data set that I talked about, where yeah, it's a very large data set, and so I absolutely think that that's a great use case for platforms like this. So, with all of these data sets, there's you apply for grants to host the data sets, and then the group that is responsible for it decides kind of which trusted research environments are going to be the hosting environments for that particular data set. And for all of us, like that's kind of Terra and this All of Us researcher Workbench. For the UK Biobank, it looks like that's going to be DNA Nexus only. So I think you would have to go to DNA Nexus to analyze it, but kind of all of the same ideas. They have expressed some, that they might consider other platforms as well, and that's certainly something we're pushing for, because we would love to make UK Biobank available in Terra as well.
I think it'll be a wonderful value add to both Biobank and to Terra if we could have those we could have this marriage happen.
Completely agree.
My other question is really just about open source, and I'm going to take us a kind of couple of decades back here. And I really wanted to talk about the sort of like two different approaches to open source. So Eric S. Raymond described this kind of framework in his famous essay, The Cathedral and the Bazaar, where essentially you can have a bazaar, which he was talking specifically about the Linux kernel development, where it's not just open source, but also open contribution. So essentially all of the communication about the project and also all the things like how to run the project, how to set it up, this is all optimized for as many contributors as possible. And obviously that creates its own challenges, and perhaps if we had more time with Wolfgang, we could have really dived into this with Bioconductor. But we can dive into this with Terra, because Terra seems to be a bit more of a cathedral. So as Dana's question pointed here earlier, it's kind of difficult to run Terra as a whole piece. It's spread across multiple GitHub repositories. You kind of need this kind of internal sort of broad knowledge to be the data, the kind of internal knowledge from the Broad Institute to be able to run it. So could you just tell us a few words about this whole distinction, and where do you see this project evolving, and what are some of the benefits and challenges of this approach that you've chosen to build Terra?
Yeah, absolutely. So I think about that, cathedral versus bazaar model a lot. I think it's a really good way to think about it. And there's a lot of huge advantages to the bazaar model, and I really believe in it. However, when you're building a platform like Terra, it takes a lot to set that up successfully, so that you're receiving all of these contributions in a way that is still secure, doesn't add any security loopholes, and you really need like a whole org just thinking about the governance of it. And in this case, because of kind of what the product is, it's not a product that lends itself particularly well to that model. However, there are parts of it that do. So the analysis tools, for example, are something where I think we certainly want like a wide range of contributions. We want to make it as easy as possible for people to add different analysis tools to the platform. That's something that we have invested a lot in recently, is a whole infrastructure for people to add kind of third-party tools. Similarly, kind of different ways of exploring the data that are in Terra, and like different kinds of data explorers. That's another area where we think you could have a lot of different contributions from different people that would make that easier. For the core components, we have invested in partner institutions being able to add contributions. So, I mentioned earlier, this is a collaboration between Verily and Microsoft. So we do have developers at both Verily and Microsoft who contribute code. But you're right. Right now, you have to go through like a security background check and a lot in order to actually be able to contribute code directly to the platform, to those core components. So that part is very much the Cathedral model, and that's really just kind of about the effort that it would take us to set up all the governance needed to do otherwise.
I see. Interesting. It's just you're almost saying that because it's almost like because of those restrictions, we have to build it this way. So it's almost like the product's driving you in that direction, in terms of, it's almost like the product is driving the community around it, and the way that developers interact when building it.
Yeah, I mean, I think it would still be possible to set it up in a different way, just maybe more challenging. And I think because of what the product is, it lends itself a little bit more to the Cathedral model.
I see. We've got another question from the audience, which is really actually really a question that can be split into two. So the first question is really about how do you prioritize things. So how do you decide which data sets are going to be available in Terra first? Would you be able to address that? Would you be able to address that question?
Sure. Yeah. So, I mean, a lot of that is, I'd say it's a combination of, data sets that are really scientifically interesting and important. So, the All of Us data set, for example, is super important because of how diverse it is, as well as the size of the data set, as well as, just practically kind of which projects we're part of. And where we have kind of partner groups that are generating the data that we can work with to make them available on the platform. But we want to make as much data available through the platform as possible. So we don't often say no to a data set. I think if you go to the data sets library, you'll see it's pretty comprehensive. We really are trying to make as much data, as much as scientifically interesting data available through the platform as possible.
I see. That makes a lot of sense. Claire, my final question here is really going to be just about a fundamental difference about the Broad and about Terra to some other products in this space. So as you mentioned, DNA Nexus, 7 Bridges, LifeBit AI, these are all commercial companies who are attempting to do similar things, but but which have this kind of freedom of being able to like almost do whatever they want, because they're usually governed by a small number of people, and they can do things like acquire companies, sell off assets. And obviously Broad is not a commercial institution in a sense. It's not a company. It's not about revenue. It's driven by some other metrics. So could you just talk a little bit about sort of the difference here, and what makes it special, and how it gives you competitive advantage, perhaps, especially being associated with Harvard Medical School, with MIT, with the kind of Boston ecosystem that's really lively.
Yeah, absolutely. It's a great question. It's funny that you say that Seven Bridges and DNA Nexus, because they're a for-profit, can do whatever they want. I actually think of it generally as the reverse, where because they ultimately they have profit motives at the end of the day. They are somewhat restricted by needing to make sure that what they do is profitable and the thing that is going to maximize profit over time. Whereas because for me, I know when I first joined the Broad, I came from a software startup, and that the goals that you have as part of a small startup provide a lot of clarity, and give you a lot of clarity around exactly what you're trying to optimize for. But it's also a little bit restricting. Whereas I think at the Broad and working in the data sciences platform, really funding still matters, but really the thing that we're optimizing for is impact, and impact on research. And I think that's very freeing in many ways, because it gives us a lot of freedom to define impact, and to go about it in a way that we think makes sense.
Okay.
And I think there's also just a huge advantage being at the Broad Institute, where we're working right alongside
where the science happens.
Where the science happens, and where we're working alongside some of the best researchers in the world. The project that I talked through with the CDC and COVID data, that wouldn't have happened if we weren't working closely with the Sabeti lab. And there's many other we actually really like for most of our projects to be collaborating with scientists like that, so that we can provide the software infrastructure, and they can provide the scientific analysis and help make sure that we're solving the right problems and doing the thing that does have the best scientific impact and the best impact on research.
Okay, wonderful, Claire. This is really great and somewhat unexpected answer. I see the thing from a different perspective, I suppose, but it's great to see this different perspective as well. We will have to move on into a jobs fair and a networking session. So Claire, thank you so much for a wonderful talk and for answering all our questions. And I'm looking forward to interacting with you in the future.
Thank you. Thanks very much. Thanks for having me.
Yeah.